Today, I'm going to present the identification of transthyretin tetramer kinetic stabilizers. So, I am Dr. Srinivasan. I'm a postdoctoral research associate at Rensselaer Polytechnic Institute. So, let's have a brief history of this transthyretin amyloidosis. So, it's a kind of disorder characterized by the accumulation of amyloid fibrils, which are misfolded proteins that can deposit in various tissues and organs. When the misfolding happens in the nerve tissues, it is called neuropathy. And when the misfolding of proteins happens in the cardiac tissues, it is called cardiomyopathy. So there are two types of amyloidosis. One is hereditary amyloidosis. Another one is wild type amyloidosis. So before going into the topic, let's see how this transretinol plays a role in the visual cycle. This all trans retinol is our dietary retinol. So it gets converted into the all trans retinal ester using the enzyme called LRAT. And using the with the action of RPE65 isomerase, there is a isomerization from trans to 11-cis retinol, and on the action of 11-cis dehydrogenase, and this is the retinal pigment. So which on isomerization, it gets converted into all trans retinol. And once the all trans retinol is formed, the transporter protein ABCA4 converts that into all trans retinol, and then the cycling and the visual cycle continues. But for some reason, this all trans retinol, when it is not cleared from the retinal pigment, so what it happens is it forms it forms an A2E, N-retinal A2E. So this reduces the rate of A2E and lipofuscin and slows or halt the progression of GA. So for the all trans retinol to travel into the visual cycle, it cannot travel alone. So this is because the molecular weight of this all trans retinol is very small, and thus it gets excreted into the urine. So it has to form a complex between the RBP4 and the TTR. So when the all trans retinol forms a complex, it can travel through the visual cycle. So let's see how it works actually. So this we have designed the bispecific RBP4 antagonist and TTR kinetic stabilizers. So as I mentioned earlier, all trans retinol, when it binds with RBP4 and TTR, it goes into the visual cycle. But in order to clear away, stop the excess production of this all trans retinol, we have designed a selective RBP4 antagonist. So when the selective RBP4 antagonist, it binds with the RBP4 and it gets cleared out in the urine, and the TTR remains. And this ATTR is the cause for this transthyretin amyloidosis. So we have also designed a molecule that can not only act as an RBP4 antagonist, but also acts as a TTR stabilizer. So it not only binds to the RBP4, but also binds to the TTR. So when this TTR is bound, it prevents the formation of this ATTR. So let us look at the structure of the TTR tetramer. TTR is a tetramer. It is a dimer of a dimer, and it's a mono and it forms a dimer, and it forms a monomer. So all these three processes are in equilibrium: tetramer, dimer, and monomer. But for some reason, and due to age or due to the genetic modification, once this monomer is formed, it no longer goes back to a dimer, but it misfolds. When the misfold happens, it forms an oligomers, and this oligomers keeps on forming one or the other, and it forms this amyloid fibrils. And this is the cause for this polyneuropathy and cardiomyopathy. So kinetic stabilization of this complex, TTR tetramer complex, can prevent the misfolding of proteins and thereby prevent this TTR amyloidosis. So this is the structure of this TTR. So this is the T4 binding site, and these A, B, C, D is the TTR tetramer. So here you can see. So this this is the yellow color is the RBP4, and this green color is the retinol. Here it forms the RBP4. It forms a complex with the retinol, and this whole complex forms a complex with the TTR. Now, when we design an RBP4 antagonist, what happens is these two loops doesn't come favorable for this binding, and therefore it can bind the formation of this tetramer. So in the market, tafamidis has been the approved drug for the treatment of this TTR. Some of the phase 3 clinical studies shows that only 30% of the patients react to this acoramidis. So phase 3 acoramidis phase 3 clinical trials of this acoramidis are still going on. So approximately 30% of the patients do not respond to tafamidis, indicating there is a need for exploring additional classes of TTR kinetic stabilizers. And this is the reported compound. This is AG10, and based on this, we have designed our compound. So the thing is, we wanted to keep all these favorable interactions of this pyrazole and these two methyl groups, this fluoro and this acid group. So what we have done is we have installed a conformationally locking group, so that to bring a favorable entropy and the activity. So this is our compound 14. So this is the Mo model. See how it fits into the binding pockets. So there is so it consists of an acid, an ester group, and there's two methyl groups. So the alkyl side chain, it binds with the HBP4, and this acid binds with the lysine residue. And these are the activities of our compound. All the activities, you can see this RBP4, the reported compound tafamidis is 410 nanomolars, and AG10 is 160 nanomolars. And our compound, it's a 14, it's almost equal to this AG10. So the thing to be noted in this activity is all these are all active in TTR, but they are not active at RBP4, meaning that it doesn't prevent the formation of the complex formation of RBP4, but it could prevent the formation of the RBP4 TTR complex. So you can that is denoted in the IC50 values. Here you can see the tafamidis, it is called 980 nanomolars, and our compound is around 845 nanomolars. So we have done with different groups actually by removing the fluorine, by installing an ester group and methyl group. All these compounds are all active. So here it's just a walk around of fluorine. We have kept fluorine in the ortho, meta, and para position. So all these, they are quite active at the TTR, but they don't show any activity at the RBP4. So then we have done some structural activity relationship by changing the groups in the mid region, by changing the piperazine region with this bicyclo compound and this spiro compound. But out of all these things, this compound number 14, the piperazine, shows the best activity. And then we have done the head group region, the structural activity relationship in the head group region. We have tried with pyrazole, oxazole, tetrazole, and the methyl group. So of all the functional groups, pyrazole seems to be the best of the lot. And then we have substituted by classical acid isosteres by amide and the tetrazole. here you can see in the compound 54, it's the tetrazole is still active at TTR, but unfortunately it's not active. It is not capable enough to disrupt the RBP4 TTR complex. And here we have done these studies, like we have done the solubility, microsomal stability, liver microsomal stabilities at remaining at 30 minutes. We have tested at human liver microsome, rat liver microsome, mouse liver microsome, and monkey liver microsome. So out of all this, you can see that all these compounds have the solubility of 200 less than 200 micromolars. So it's a very low microscomal intrinsic clearance, and which has been reflected in the human microsomal liver stability. And none of these compounds have shown a significant CYP inhibition. So when a compound is showing a significant CYP inhibition, so it no longer reaches the target. So fortunately for us, it's the very less amount of CYP inhibition is there, which is also reflected in the percentage plasma protein binding. And here we have tried with the acid aggregation assay. So this graph B, it shows the amount of tetramer formation. So we have done at different conditions. One is a TTR at neutral pH, and the other one at acidic pH, and the other one acidic. The compound, the graph C is acidic pH plus 14 micro plus compound number 14, and pH 4 at and the reported compound tafamidis. So when you look at the graph B, so you can see the amount of the tetramer doesn't break at neutral pH. Therefore, we see a very less amount of high molecular weight oligomer. So, but at the acidic pH, this tetramer falls apart, and therefore you see a higher concentration of the amyloid fibrils. In the graph C, it is still at the acidic pH, but since our compound was able to stabilize the tetramer, we can see the amount of the misfolded oligomers decreases significantly. The same is the case with the reported compound, which is shown in the D at acidic pH at 50 micromolars of tafamidis. Still, it shows the same amount of the reduction of the tetramer. So by this graph B, we can say that our compound is able to decrease the disformation of the tetramer, and here we have monitored the formation of the dimer. So at acidic pH, at neutral pH, you see a lot of dimers, and acidic pH, the dimer decreases. And but in presence of our compound, the amount of dimer increases. And compound D, it shows we have calculated the amount of monomers. So at neutral pH, you see a lot of monomers. At acidic pH, the monomer breaks into the misfolded oligomers. Therefore, there is a decrease in the concentration of the monomers. Here you can see some monomers in presence of our compound C and compound D. Compound D is the tafamidis. So all we can say is our compound is able to stabilize the tetramer. We can see some dimer and we can see some monomer. So this is what exactly we want to see. So we can say for surety that this compound is able to stabilize the tetramer kinetically. So here we can see the graph at F. So this is the graph is F is the combination of all these A, B, C, D, E graph actually. So here we can see with the AG10, so the red colored lines. So there is a decrease in the concentration of the serum RBP4 level concentration. And same goes with the tafamidis as well. So 14A, our compound was also able to decrease the serum RBP4 level concentration. So here we have done the PK-PD correlationship between. So here in the first graph A, so at 2.5 hours, you can see there is a decrease in the serum RBP4 level concentration at the same time period at around 2.5 hours. So here you can see the increase in the drug concentration. So there is a nice correlation is observed. So whenever the drug concentration increases, there is a decrease in the serum RBP4 level. The serum RBP4 level concentration decreases. So which is what exactly we want to see, the exact PK-PD correlation. And to summarize, so we have developed a potent TTR kinetic stabilizers. It was able to disrupt the RBP4 TTR complexation. The stabilization of TTR aggregation assay observed in the in vitro SDS gel, and it lowered the RBP4 level in systematic circulation of mice. Studies with 14 shows in the ABCA4 mouse model are still ongoing. So I would like to thank Professors Dr. Costa Petrukin from Columbia University and Dr. Christopher L. Siofi from Albany College of Pharmacy. So he is my research advisor, Dr. Christopher L. Siofi. And I would like to thank my team members and also the NIH for their funding, National Eye Institute, and also the National Eye Grants and Research to Prevent Blindness. I would like to give the acknowledgements to all of them. And thank you for your time. Thank you.